Hello guys, how are you doing? Let me introduce myself. My name is Ahmet and I will give sound for this presentation. This is biological chemistry for second course, and today we'll talk about biochemistry of lipid metabolism. The first, metabolism and function of lipid. We need to consider these two most important topic. Lipids of the human body include compounds that differ significantly both in structure and function in a living cell. These heterogeneous substances are combined into the class of lipids based on their common property, hydrophobicity, and solubility in water of the entire or a significant part of the molecule. A number of pathological conditions are studied with lipid metabolism, such as obesity, cholelithiasis, metabolic acidosis, and atherosclerosis. The first, lipid classification. Lipid classification divided into several groups. The first one to consider fatty acids. After we'll consider fats. Triglycerols. This is food lipids, are mainly present in the form of fats. Phospholipids and glycolipids, complex lipids. Steroids, the most common representative in cholesterol. Eicosanoids, derivatives of arachidonic acid, and perform regulatory functions. The first, we need to consider fat digestion. Daily requirement for fats is 50 to 100 grams, and the oral cavity lipids are not digested. Saliva does not contain the enzyme lipase that break down fat. Lipids are also ingestible in the stomach of an adult, neutral slightly alkaline environment. In addition, the fat must be in an emulsified state. Children have their own characteristic. Bile contains conjugated bile acid, including glycolic, and taurocolic. In addition, bile contains phospholipids and cholesterol. Absorption of fat digestion products. Fatty acid absorption occurs in the proximal portion of the small intestine. Fatty acids with a short carbon chain and glycerol are freely absorbed and enter the portal vein and blood. Fatty acids with long carbon chains, monoacylglycerols, together with the bile acids and phospholipids of bile form mixed micelles, and then all components of the micelles penetrate into the cell of the intestinal mucosa. Digestion and absorption of phosphoglycerates and cholesterol. Phosphoglycerates are catalyzed by the action of special phospholipase enzymes that are found in pancreatic juice. They are divided into phospholipase A, A2, C, and D. Cholesterol not digested with food, 0.1 to 0.3 gram intake. The synthesis of fat in intestinal cells. In the intestinal cells, most of the products of digestion are again converted into triacylglycerols. First, fatty acids are activated to form acyl-CoA, and then acyl reduced are transferred to monoacylglycerol with the participation of transacylases. Transport lipoproteins. Transport of fats and other lipids is carried out as part of special lipid particles. Lipoproteins are multimolecular structures. They are spherical particles, the surface part of which is formed by a monolayer of oriented to phospholipids and proteins. The blood contains several forms of lipoproteins. The main ones are chylomicrons, very low density lipoproteins, and high density lipoproteins. Lipoproteins differ in composition and content of lipids and proteins. Fats synthesized in the intestinal cells from the products of digestion of dietary fats are included in the same cell in the LP kilomicron, a little bit LDL. Lipoprotein lipase. In the media of the capillaries of muscles and adipose tissue, there is the enzyme lipoprotein lipase, which hydrolyzes lipids of lipoproteins. Lipoprotein lipase is synthesized in adipocytes cell of the heart and skeletal muscles, and some other organs. Fatty acid metabolism. Fatty acids are the simplest lipids in structure. A variety of fatty acids are found in human lipids. The vast majority of body fatty acids have an even number of carbon atoms. Human adipose tissue contains the largest amounts of oleic, palmitic, and linoleic acid. All transformation of fatty acid in cell begin with the formation of acetyl coenzyme A. Acetyl coenzyme A is formed as an intermediate product in both oxidation and fatty acid synthesis. The inner membrane of the mitochondria also contains the enzyme carnitine acetyltransferase, which converts cell carnitine into acyl-CoA and carnitine. Here, carnitine again returns to the cytoplasm and acyl-CoA undergoes beta oxidation in mitochondria. Beta oxidation includes two reactions: the hydrogenation 1,3 and hydration and thiolysis reaction. The overall results of the oxidation of palmitoyl CoA can be represented as follows. Biosynthesis fatty acid. In addition to dietary fats, the source of fatty acid in the body is their synthesis from glucose. The precursor of fatty acid during their synthesis in the body is acetyl coenzyme A. The same substance that is formed during beta oxidation of fatty acids, despite the fact that all beta oxidation reactions are reversible, they are not used for fatty acid synthesis. Stage of fatty acid biosynthesis. Stage first, formation of malonyl coenzyme A. Second stage is biosynthesis in the palmitin sinus complex. Formation of malonyl CoA. It is formed from acetyl-CoA, which is formed in mitochondria in a common catabolism pathway and transported to the cytosol by a shuttle mechanism. Second stage, palmitin synthase. Palmitin synthase is a multifunctional protein in catalyzes a series of reactions. Palmitin synthesis consists of 6 enzymes associated with the so-called acyl-carrying protein. This protein is thermostable, has 2 free sulfhydrogen groups, and is involved in the process of synthesis of higher fatty acids at all its stages. Then the entire cycle of reaction is repeated. Let us assume that palmitic acid is being synthesized. In this case, only the first of 7 cycles is completed with the formation of butyryl APB. Fatty acid synthesis is completed by cleavage of sulfhydra ACP from ethyl ATP under the influence of the enzyme deacylase, for example, this reaction. The overall equation for the synthesis of palmitic acid can be written as follows. Thank you guys for attention.